today we have Dr. Sarah Keller from the University of Washington in Seattle. She's a fellow of the APS and the Biophysical Society. She studies self-assembly, soft matter, and membranes. And so we're going to learn a bit more about that today. And please make sure to have, if you have any questions to ask Sarah, and let's make sure we have questions from students. All right, thank you.
Thank you. I'm surprised, I'm delighted by how many people are here on the first week of your term. So thank you very much. I feel very honored. I know that on the first week of term, sometimes it is hard to get back into things. And so I am hoping that you'll ask questions. I don't feel any need to have gotten through some amount of material by the time we get to the end. The whole point is to really more interact with you. I also know that this is a MRSEC seminar, and that umbrella of MRSEC can be really super broad. So I don't know exactly who you are in the audience. There are physicists, there can be chemists, there can be biochemists, there can be material scientists. And so I'm hoping that you'll ask your question no matter what way you approach it from. I might not know the answer from all those different angles, but hopefully I can tell you who might know and then what papers you might read from that. Okay. So I'm going to tell you a story that has a cast of only 3 characters. There is a lipid that has a high melting temperature. Okay, so a lipid is an amphiphilic molecule. It has a hydrophilic head group, hydrophobic oily tails, and they make membranes. And there are many different structures of them, and I'm going to really simplify it in terms of its material properties. There is one of those molecules that has a high melting temperature. That means that its chains are really long and saturated. So you could think of like butter as a saturated fat. There's a lipid that has a lower melting temperature. It has carbon chains that are unsaturated. So you could think of something like olive oil in terms of that case. And there's also a sterol. We tend to think in our bodies in terms of cholesterol. I'm going to tell you a story about yeast. They have a different sterol, ergosterol. But all of the intuition that you might have in your kitchen about butter and olive oil and cholesterol, you can transfer over to this knowledge. Okay, so we have 3 materials. They happen to be materials that show up in cell membranes. And I could also take my physicist picture of a cell and think about making a really simplified version that has no proteins. So I study both extremes of this case. I look at living cells, and then I also look at completely artificial membranes that have no proteins whatsoever in them. There's a beautiful, huge range in between that I don't study. Okay, so if we think about an individual lipid, in these cellular contexts, it is in a liquid phase. And that means, one of the things that means is that it is able to diffuse everywhere within that surface. Okay, so it's this little person, big head, 2 tails, walking around on the surface. There are many instances of that particular being on both sides of the membrane. And just like people, what makes this really interesting are the interactions between the different individuals in the case. Sometimes we all like each other better than we like other people, and we'll tend to associate with each other. If those interactions are strong, either the attraction or the repulsion are strong, then you can get a phase separation in the system. I'm going to tell you a side note, which is that so far, only 2 coexisting phases have been seen, and even when you have n components of the system. So Gibbs phase rule says you could get many. Even if you have a cellular context, which has a gazillion components and all the different proteins, so far, only 2 coexisting phases have been seen.
That's robust, Sarah.
Oh, that's a great question, but I'm not going to let you ask that because the first question has to come from a student.
I didn't know that no one had asked.
I know, that's okay. But I will answer it. It's not right now. I'll wait. See, think of how uncomfortable you're making her. Don't you want to relieve her stress and ask a question? Okay, this is temperature dependent. In this particular system, those two coexisting phases go away at high temperature. There's uniform mixing up at that high temperature, and there's a distinct transition at which that occurs. We can draw that on a phase diagram. So typically, some of us were talking earlier about like how we might not have liked undergraduate thermodynamics as much as maybe we grew to like it later. So here's a phase diagram. If you open up an undergraduate thermodynamics textbook, you'll see a phase diagram like this, where we have temperature on the y-axis and the composition, so the amount of some component over here on the x-axis. At a high temperature, there's one uniform phase. At a low temperature, there are two coexisting phases. And where that line is of the transition depends on the particular lipids that are in the membrane at that particular point. And there's a really long history of mapping liquid-liquid phase diagrams from model giant vesicles. So our group, many other groups as well, have done this for a really long time. And here's what that looks like in the microscope. So the system that we do that in is it's called a giant unilamellar vesicle. So what giant means 100 microns in lipid land. Unilamellar, one thin sheet. It has water on the inside, water on the outside. And in this case, it's taut. So it has only enough membrane to cover all of its volume. It doesn't have extra area in it. And it's going to start at a high temperature. And this one's so much better from the back of the room. So I'm going to come back.
You can turn the light on.
Okay. Oh, right.
The other one.
Oh, you have the best seat in the house. Okay, so what you saw start off at a high temperature was uniform and gray, and then plunge down to a lower temperature. Little dark domains nucleated. They are diffusing around on the surface of the vesicle. Oh, I wonder if the people online can hear me from there. And then you can tell that the white is liquid because there's fast diffusion of those. This is approximately real time. Fast diffusion of the dark domains on top of that. You can tell that the dark are liquid as well because when two domains collide, they coalesce really quickly. If we had the patience to watch this for 10 minutes, 20 minutes, depending, we would see that we would only end up with one bright domain and one dark domain. So same thing as when you have oil and water, salad dressing, you shake up the bottle, you end up with little domains. They run around with Brownian motion. In that case, there's some creaming as well. And then domains collide and coalesce. You end up with only two. Any student clarification questions? Are you a student?
Yeah, I'm a student. How fast do they start, like, they separate from when you?
Excellent question. So we're doing microscopy. That means I only start to see them once they're about a micron in size. But I will say that it looks totally reversible. And as fast as we go, as we don't have super fancy temperature quenches. But as soon as we go through that transition and then it's reversible the other way around, we see the transition immediately. But I mean, I'm sure that there's some interesting nucleation behavior, but we don't have the resolution either in time or in space to see it. Now there was a question about how robust is it that you only see two coexisting phases in a membrane that has many components. If we're talking about micron-scale liquid-liquid phase separation, so far, I only know of cases in which there are two coexisting phases, but something could be discovered. Absolutely, it's absolutely allowable, and it's kind of surprising that there have only
And
been
the
two.
porcelain also happens in sort of the same way.
Yes, yeah, even in these very complex systems, as long as the membrane is taut.
Mm-hmm.
Yeah. Oh, here, I'll go back while there's still questions. And so you can watch the
You
movie
see
again
the
if you want.
same composition of both sides of the membrane?
Do we,
know.
oh, you mean of the two leaflets?
yeah.
Ah, in these particular systems, yes. There is the same lipid composition in both leaflets. In fact, it's rather challenging in an artificial system to get asymmetry in the membrane. And that's a whole other talk. I could just talk your ear off about asymmetry because it's really beautiful. Thank you so much for the questions. Great, yes.
So does the diffusion coefficient follow the size?
Yeah, so the diffusion coefficient is going to depend, that is another talk. And I'd love to talk to you about that too. So the diffusion coefficient in this case depends on two different things. There are two different viscosities in the system. There's the viscosity of the surrounding fluid, and there's the viscosity of the membrane. The viscosity of the they have different dimensionality because it's a two-dimensional viscosity of the membrane and a three-dimensional viscosity of the fluid. So when a domain is traveling through the membrane, if it's in a very viscous membrane, then it has to, like when you're at a concert with all your besties, right? And you have to like all push through the crowd together. Then it's going to experience this very large diffusion in that direction. It's going to be dominated in that case, and then it does not look just like Stokes-Einstein. But if the membrane viscosity is not huge and the domain is big enough, then it's taking a bunch of fluid with it, and then it's just acting like a big ball. And then it just looks like Stokes-Einstein. So the relationship of going as 1 over radius versus log 1 over radius, there's a crossover in that. Sorry to like go down this total decol on that one, but I could talk about that for a long time. Yeah.
How does the coalescence behavior change in a slide membrane?
Oh, I'm going to show you a little bit about that, but let me just say super briefly in case we just don't get to it. So if the system has excess area, then these domains, movie going to go? Yeah, it's going, right? Not yet. There we go. Okay, then these domains end up bulging out of the membrane. So they have different bending coefficients because they're different materials. They have different material properties. So if you can imagine a membrane that maybe only had 2 domains, it would have like Mickey Mouse ears on top of it. And so when those ears start to approach close to each other, the membrane that's in between starts to bend. And that is like a spring, and it prevents the 2 domains from merging together. So you can get that, you don't get coarsening in that same way. You can get this arrested coarsening due to that.
From the elasticity
Of the membrane, the 2D elasticity of that membrane. Yep. Awesome. I feel like I am in the right place. Really great questions.
And sorry, is that enough? But it's not enough to kind of have a finite side, right? There's no self-limitation in that, where you will not get like a uniform kind of size distribution. There's like a big size distribution.
Right. There's a question of what causes the size distribution, and I'll show you that right at the end. And if I don't get to it, then I understand we get to talk a little bit later too. Okay. So as we go to more biological systems, right? So many years, I was really happy over here. So I'm hired in chemistry. University of Washington is super interdisciplinary. I was trained as a physicist. Then I did postdocs in a chemical engineering lab and in a chemistry department, and I was hired in the chemistry department, but I don't make any molecules. So I buy all my molecules from Avanti Polar Lipids, and they're all just stored in the freezer, and then I just mix them in different ratios. And then there's this question of what now happens as you start making this system more biological. So it turns out that you can get this same sort of really huge micron-scale domains within cell membranes. So I think that especially from those of you who have come from a biochemistry standpoint, even your textbooks now might start to talk about rafts, and they're usually characterized as submicron domains in a membrane. These are really big, micron scale. Okay, and so we have studied it, and then I got really excited about it by initially reading a paper. And I read a paper about the yeast vacuole. So I am still new to yeast. I'm sure that many of you know a lot more about it. Let me tell you a little bit about the vacuole. It's part of the endocytic pathway. And the reason I got super excited about it was because I read this paper by Alexander Tolmay and Will Prins back in 2013. And like as a scientist, you're super busy. How do you read a paper? You start flipping through the pictures first, right? And I'm like, polka dots in membranes. I study polka dots. They study polka dots. But are they the same polka dots? Like lots of things make polka dots. So I want it to be a liquid-liquid phase separation. Is it really something? Oh, this paper is so gorgeous. One of the things that they did was they looked at 14 different endogenous labels of proteins, 3 different lipid probes. No matter how they looked at the system, they only saw 2 coexisting phases in their system. So it was really robust as far as we know. When did they see these domains show up on the membrane? Well, they saw it in what's called the stationary stage of the yeast. So here's how yeast grow. Starting at the left, there's a yeast that has a lot of sugar. It's very happy. It's making a lot of buds. Its vacuoles on the inside, there are many different vacuoles. And then the yeast starts running out of glucose to eat. It undergoes a shift, all the vacuoles merge, and then they break out of polka dots. Really cool. They don't tend to all merge because that membrane is not taut. It has excess membrane there. Okay, what am I going to tell you? Maybe I won't get through it all. That's perfectly fine. First, I'm going to tell you that those domains in vacuole membranes in living yeast, right? So yeah, they don't have as much glucose as they would like. When do any of us? But those domains are huge. They're still in living yeast. If you put them back in glucose, they're perfectly happy. They are due to a reversible phase transition. The second thing I'm going to tell you is that yeast regulate the temperature of that transition. They really, really care about that transition. It's not just like, oh, this happens to happen. The yeast really care about it. And in order to achieve that transition, they must be changing something in their system. It's got to be something with chemistry. It's got to be something in the membrane. Turns out that they are changing their membrane composition, especially they're probably changing the proteins as well. They are at least changing the lipids. And sometimes we get a particular length scale in the system. We don't always know why. There are several theories, but no current theory completely explains why. Okay, here we go. The stars of this story are not me. The stars of this story are Chantal, Caitlin, Scott, Glennis, brother, sister team of awesomeness, and our collaborator, Alex Mers, who's in the biochemistry department. So they came in all with like really different interests into our program. You don't have to know what you want to do in grad school to be admitted to the program. So Chantal knew she wanted to do something with cells, probably not with my group, and then ended up joining my group. Caitlin initially came in saying she was going to do inorganic chemistry. Scott and Glennis, I think, both came in thinking they were going to do physical chemistry. That's a little bit unusual for my lab. And then Alex is in the biochemistry department, which is a different department than chemistry at the University of Washington. It's in the medical school, which is just across the street. So it's literally 7 minutes. I know because I'm often late for meetings. I'm like, 7 minutes, run. Look at you, Alex. Okay, here's the experiment. We're going to change temperature over time. You see a bunch of dark dots and a bunch of bright dots. The dark spots are where there are dark spots on that vacuole membrane. Okay, so as we go up in temperature, the yeast are for this is only looking at one cell. Okay, so we're going to eventually look at many cells, but this is only one cell. It's growing. It's been grown at 30, And then we're going to take it from its growth temperature to a higher temperature briefly, and then sawtooth our way across, okay? So initially, there is one domain. This is in the stationary stage. It does not have a lot of glucose, right? So those vacuoles have merged. It has domains. And then bring up the temperature, the domain goes away and comes back, and goes away, and comes back, and goes away, and comes back again. And it is totally reversible at the same temperature every single time, even though it is a living system. Yeah,
What are we looking at? Is this a protein marker or lipid marker?
that's a great question. This is a endogenously labeled VPH1 protein. It lives in the vacuole membrane. And its role is to, one of the roles of the vacuole is to maintain pH, and it's one of those proteins in that membrane. But you could do it like Tolmay and Prinz showed. You can do it with many, many different endogenously labeled proteins. Good question. So we already know the first one. Second one, yeast actually care about this transition. So the stars, oh wow, the superstar of this is Chantal. And we started asking the question, instead of just looking at one cell through time or a few cells through time, let's look at thousands of cells and build up some decent statistics. And so what Chantal did was wrote a program in order to look at a whole field of those yeast. And then her program picks out those yeast from the picture, puts them all in a file, labels them with the file names, but then scrambles them and shows them to her in an order she doesn't know, and says, does that one have domains? And she says, yes, no, or I can't tell from it. The number of I can't tells is very small. And then later, so it's a semi-blind procedure, and then later she actually gathers the data and then plots them here for you. So what I liked is that it's really semi-blind, so she doesn't actually know what things are going on. The yeast are grown at 30. And then I've already told you that at low temperature, they do have domains. So this is what we're looking at on the y-axis, the percent of membranes of vacuoles that do have domains. Okay, so we already know at low temperature, they have domains. So up at 100. And then at high temperature, this should be down at 0, but we don't know how it goes from there to here. How does it happen? So hundreds of cells in each experiment. So each point is hundreds of cells, and it's super sharp. It's just this cliff. If you had shown me these data, I would, without me knowing what system, I would have said, nice artificial vesicles you made. Nice, extremely well quantified, beautiful, pure system you have because you got such a sharp transition. But this living system of thousands of cells is doing the same thing. I was really blown away by this. Now, there's an offset here, right? I already told you that they undergo that transition above their growth temperature, right? So when they're at their growth temperature, they're happy to have domains. They want to have domains. What sets the temperature? Like, is it that there's some protein in the membrane that's denaturing and it happens to denature at 47 reversibly somehow, though that would be hard. Or is it the offset? That's important, and I kind of gave you a clue before about what that is. So to test that, Chantal grew yeast at different temperatures and then found that the temperature of the transition scales with the temperature of the system. So this is a little freaky to me. Like, if you have temperature versus composition, and I already told you that at high temperature, there's one uniform phase, and at low temperature, there are two coexisting phases, and it looks something like this. then for the physicists in the audience, up here, there's a critical point. And that means that if you're a little bit above the critical point, then you can have fluctuations in their particular size. And so the system could be like somehow measuring as you get closer down to that critical point, how far you are away from the critical point. But if you're coming from below, how does this system know how far above it the transition is? I don't know. I don't know. I have no clue what it's measuring, which is super fun. Yeah.
So maybe I'm completely wrong, but I guess what you're saying is if I plotted things as T minus the growth temperature, they would all fall on top of it.
Yeah, approximately, yes.
So why should I think that there is the T axis itself?
Oh, sorry. And where, oh, I thought I saw different colors. Awesome. Where now this is the transition if you grow it at a particular temperature.
Right, so I'm
And
saying.
then the next one, the next temperature of growth,
Right.
but the offset between
offset is from
T
the growth
growth
temperature.
and say T mix
It's the same.
is approximately the same.
So why can't I think of the y-axis as being T minus TG?
Totally can. Absolutely. But from a standpoint of like, what is the cell changing in order to accomplish that? I don't know. Or I have hypotheses. But yes, you're totally right. That's the physics way to think about it. I saw one, yeah. Did you have, oh,
You keep it at high temperature for longer, but always reversible.
yes, the question I hope that you could hear at home. If you keep it at high temperature for longer, does it shift? So we do know that if you keep it at high temperature for the whole growth cycle, right? You start over and you take them back to the log stage and you grow up these cultures again. Now at a new temperature, it does shift, but that's days. So where's the time in between? We can take our system, so here's temperature versus time, where this is about 1 hour. And we haven't done more than this yet. That'll come. And so like here's T mix of the system. And we start to at time 0, just below T mix, and then go boop, just above T mix, hold it there for an hour with the light off, and then go boop, back down again, and we still go across the transition. So we know that at least over a time scale of an hour, this transition temperature is not changing. To answer your question? Yeah.
Am I seeing that some of the curves are smoother than the sharp? And
Absolutely, yes. And I'm not exactly sure why that is, but there are some confounding variables. One confounding variable is that this particular yeast, so this is just cerevisiae, likes to grow better at 30. And so
Yeah,
now
so-
we've taken it to some temperatures, some growth temperatures where
So
it's a little bit slower at
crazy idea
growing.
coming from a stat mech person if there's a field,
Oh, yeah.
which is depending on when you're growing the, and I don't know what that field is for
Right.
a living system. But I've seen this kind of thing in lots of non-equilibrium systems where I don't understand what the field is.
Right,
But it's acting like a field to smooth out the transition.
what I don't know, would it make a difference to you if for this particular system, we can't go to higher temperatures, but there are yeasts that grow at lower temperatures. If
Yeah,
it were
if you
best here,
showed
but
that
then
there
up
is a-
here, it got broader as well.
Yeah.
Oh, sorry, then would that change?
Or if you, so that's, I'm thinking if you can try to collapse the curves on top of each other
Mm-hmm,
with just a shift in T,
mm-hmm,
then there is no field.
mm-hmm, right,
But
right,
if
right,
you
right.
can't, then there is an additional parameter that depends on the growth temperature.
I guess what I was asking is, does it matter if it's re-entrant?
Probably not,
Okay.
yeah. It would be different from, a magnetic field, but yeah.
Like how happy are these at higher temperatures? Like what is the fitness?
Great question. We worried about that. Because as you might have seen from that first one, like we make these brief excursions up to like 50. And most, like the kind of the general knowledge is yeast aren't so happy at 50. So what we did was we took a culture and then we split it. And half of it we did under the microscope with torturing it with temperature. And then half of them we did the exact same protocol of temperature and then grew up the colonies and compared them to one that we split it in 3, in which we had not tortured them in that way, the same number of colonies. So the fitness seemed to be the same as long as we were not holding at a high temperature for a long time. But other people in the field have said like, oh, don't like let it hang out there at 50 for days. So we tried to make excursions up to 50 only very briefly.
Yeah, I guess it's just like, if you have something that's growing at 30, I don't know much about yeast, but you start something that's like growing at 30 happily, and you like increase it by temperature like, I don't know, 8 degrees or something, right? So here you have like no change in the domain. Like if your offset is like, sorry, if you increase temperature like 10 degrees or something like that, you're seeing like no change in the like structure of the vacuole, like the average vacuole.
That we can see by
That you see.
microscopy.
Right, so my question is like, is there a fitness there? It seems like there's only,
There doesn't seem to be, at least in terms of how the yeast grow, but that's a pretty blunt measure. Yeah,
I had another question a little bit similar to that. So I had another question about the yeast growing at a temperature that we know is above their optimal rate. I was wondering if you'd seen any potential, or were worried about any potential denaturation or any kind of cell death at that temperature.
right. So we definitely were, and so that's why we measure the fitness. The fact that this is all reversible and that it scales with the growth temperature implies it's not a particular protein that's denaturing. First of all, it has to be reversible, and then it would have to denature at different temperatures depending on the growth condition. And we haven't grown them at, like these grow best at 30. We've only grown them at temperatures that are below their happiest growth temperature, which is legal. You can tell I'm not a cell biologist. I talked to a lot of them. Yeah.
Sorry, one last question. So when you grow it at 30C,
Yeah.
and you look at this population average, that suggests that like all the cells have the same kind of mix.
Right. And then each of these curves is a completely different day of a new culture.
If you look at the one that's grown at, let's say 20.
Yeah.
Now you see more variability in that things, right?
Yes.
And is the variability like is there still variability if you look at single cells, like at that temperature, like, or is it just a population?
It's just a population effect.
So if you would take like the same cell and we would always have the same
Yes.
mix.
Yes. Okay, for those of you at home, in case you didn't hear, the question was, is each individual cell always reversible? And the answer is yes, as far as we've seen.
It's always the same temperature, even if it's grown at.
Even if it's grown at a different temperature. Yeah. Okay, I'm assuming I should stop at 3. So you can get to somewhere else. I mean, the time we were given was 2:10 to 3:10,
Yes.
which implies to me that there's actually, most times you actually need some like passing period, or we should stop around 3. You tell me, I will take my instructions from you. I am flexible. What would you prefer?
I don't know if there are classes here, someone from our section.
Anybody else passive?
Not these people, there can be classes from anywhere.
Oh.
You know.
In this room?
Yeah,
Does anyone have to be anywhere at 3:10?
no, but there might be classes here.
Oh, I see what you mean. Okay, we will try to stop, but at an appropriate time. But I still want your questions. It's okay if we don't make it through. Okay, how are these doing this anyway? So they are achieving this transition by changing their membrane composition, and we can measure those changes. Okay, so in terms of physics space and experimental space, I am most happy thinking about changes in temperature. That's kind of my world outlook. It's also how we do it in the laboratory. What are the cells actually doing? The cells can't change their temperature. They can change their composition of their membranes. They're changing the lipids in their membranes. So they're actually going through this transition, but it's thermodynamics. So it doesn't matter how you go across the transition. You'll always get the same transition no matter how you go across it. So it's still useful to map those phase diagrams. It's just that the actual cell may be assessing it in a different way. So to do this work, we teamed up with some lipidomics experts in Germany. They are John Reinhardt and Robert Ernst. And I really totally want to give a shout out to Candace and Ilya Leventhal. So we went to the Biophysical Society meeting and presented our results. We got this question over and over again: have you measured the lipidome? No, we haven't, because there are these technical details that make it very difficult. And Ilya and Candace were like, do you know Robert? No. And like, he happens to be at this meeting. And like took us by the hand, led us over to Robert, and they're like, you should talk. And it led to this collaboration. So it's one that I love being in an interdisciplinary field. I also really love being in a field where people are awesome to each other, right? Things are really field dependent. Different fields have different cultures. And I'm really happy where I ended up. Didn't have to turn out this way. Okay, so here's our cast of characters again. There are some lipids that have high melting temperatures. There are some lipids that have low melting temperatures, and there are sterols. It seems to be the case that one of the things that helps phase separation happen in membranes is by having the high be high and the low be low. And so, like, what's happening in these cell systems? How much of a spread do they have? Okay, so don't run. I'm going to block the door. It's really overwhelming, right? So what is this showing? This is showing ergosterol and all the different lipids in the system, even the ones that are there in really low mole fractions. I find this absolutely overwhelming and impossible to read, and frankly, a little bit scary, right? All the lipidomics people are like, oh, very interesting. I'm like, I didn't even look at. So let me help you, maybe if you're having that same feeling. The assumption that I would say I don't know anybody who didn't have this assumption, but there's probably some really bright people out there who didn't, was that the ergosterol would really change between the two systems. So like maybe I could give you a clue here. The really light gray is in the log stage. That's no domains in the membrane. The really dark gray is stationary stage, so that does have domains. How is the membrane? Only of the yeast vacuole, not from anywhere else in the cell. Only that membrane changing in that transition. Does that give us a sense of what might have made the domains happen? Okay, we all thought it was going to be the ergosterol. It's not. What is changing? The amount of the phosphatidylcholine. So that's a particular lipid. It's telling you what's up on its head and not yet what's in its tails. So let's look at that. That's the biggest change. I don't know. Let's go look at what's in that one. So if we look at only that particular lipid, you don't need to remember the name. Now I'm going to tell you about like how many of those lipids have high melting temperatures, how many of those lipids have low melting temperatures in either the log stage when there are no domains or in the stationary stage when there is liquid-liquid phase separation in the membrane. Okay, it's a schmear, as you might expect. It kind of looks like a undergraduate grade distribution. There's a bunch in the middle, and then a few really high and a few really low. It's not particularly elucidating to me on its own. But what happens now in the stationary stage when there are domains? Well, the middle has shrunk, and there's more up on the top, and there's more down at the bottom. So of this distribution, right, you've now gotten more top and more bottom in the system. And that fits with my world picture. It's It's hard to say exactly because like this is such a huge multi-component system. But if this were a ternary system in which we had high and low and a sterile, it would be consistent with being able to make liquid-liquid phase separation. Because we have more that are high and more that are low. Yeah.
If you look at some of the other species that didn't increase in total amounts, do you see a similar redistribution of chain length? Or is this just the new PC that was made?
This is only the PCs.
But for the other lipids, if you look at the same chart. do you
Oh, that's a
see-
really great question. So this is a place where we need more data. So the transition temperatures are not known for every lipid type. This is the place okay, I will admit that we are looking under the lamppost for the keys, but this is the place where there are data. And actually, I think that you could spin that in an optimistic way, especially for students. So when I was a physics student, I was like, I love physics. How will I ever make a contribution? It's all been done. And so I want to say that there are lots of places where there just aren't data yet. And we need all of you, please, to get data like this to tell me, like, if I find a particular lipid with that head group and these particular tails, what is its melting temperature? It's not known for a lot of different lipids. Okay. Sometimes the domains get smaller or they become stripes. And at the moment, no theory currently explains all of why, which I found really surprising. Okay, here's the deal. Star of this is Caitlin, in collaboration with Ilya and Candace's lab. So I'm gonna take a step back again. I'm not gonna show you yeast. I am going to show you instead, this one is a giant plasma membrane. What does that mean? It means that you take a cell stock, you're mean to it. There are many different ways to make it to be mean to it. The outer membrane blebs off. It's a real word. And then you can shake the system, the bleb falls off, and you can image it in the microscope. Okay, so that's what Ilya and Candace do. We see the same thing in our lab in giant vesicles that have only 3 components. So this all happens whether you have 3 components or a gazillion components. And what's freaky to me is that we start from here. We start from it having only 1 domain of each type. So full liquid-liquid phase separation. There are different ways to get these stripes to happen. So the Feigenson lab at Cornell, they do this procedure where they change temperature very slowly over days, and then they come back and look, and it looks like that. We've never been able to recapitulate that in our lab, which doesn't mean that it doesn't happen, just that we haven't been successful with that. Other ways to do it is to add just a small amount of lipids. It doesn't matter which type, actually. To only one of the leaflets, it's easier to add to the outer leaflet. So that's what we've done here. And so we started from coexisting domains, and we end up with stripes with a particular length scale in the system. And they're fluid. They wiggle around. But they have a particular length scale. Yes. Sorry, you can tell I'm a microscopist because I'm just like, oh, can't we just watch the movie forever? It's so pretty. Okay, can we control the length scale? Yeah, and there's a hint, there are lots of hints in the literature about how you might control the length scale. Yeah.
That video, is the membrane taut or slack? Because
It's-
there's no coalescing happening.
It's a little bit slack. Yeah, it's a little bit slack and it's a tiny bit asymmetric. Yeah. Thank you for the clarification question. That's really great. Okay, there's a lot of literature. And I will just say my strength is in the experiments. I found these challenging to read. So there's a bunch of theory. The ones all up at the top in red are theories that describe dots. The ones on the bottom are theories that describe stripes. We know that there are some of these theories that are not relevant. Those are grayed out to not distract you. And so what's left are the ones that are in black. That one up at the top by Reinhard Lepowski and coworkers are only describing dots. And so I asked him in person, it's like, could you get this? He has, as it stands, it doesn't apply to stripes. So that leaves these other two. Okay, what are these other two? Let me just like actually translate this physicsness into plain English for you. One says you couple the local lipid composition to spontaneous curvature. What does that mean? Okay, let's say that you have this membrane, and instead of it being flat, it's not exactly taut. It's a little bit slack. It has a little bit of excess area. So there's a place where they'll have a bump. Okay, so like it's hanging out, and then just locally a bump occurs. And where there's that bump, you've got a bit of bending. And that bending, there's some lipids that are easier to bend than others. So you might end up with one phase in that bent part versus the less bent part. That's all that says. Now, the devil's in the details. There are lots of ways when you write a theory to make that happen, and that's what leads to the diversity and the richness of the theoretical literature. The next one says, okay, you merely need some kind of repulsion between the molecules. So imagine it like this. So here's our membrane, and then we've got a domain in the membrane, and it's full of molecules. So is the outside is full of molecules. That boundary has what's called a line tension. And I think that you already have a lot of intuition from that, because if you think of a droplet of water, you're used to thinking about the surface tension of that droplet, right? And if the surface tension is high, then that droplet's really round, right? But then if it's low, then you can get a really wiggly droplet, okay? So same way, here we have some line tension that wants to keep the domain round, but what if all those molecules hate each other? They want to get away from each other as much as possible. Like they would like to make a line, but that makes a huge amount of line tension that's associated with that. So a balance between those is going to give you a line with some width, okay? And that's what could set that Planck scale. Makes a lot of sense, but why do the molecules hate each other? Like that's the physics. But where's the chemistry? What makes those molecules hate each other? There's a proposal that comes from lipid dipoles. So let's try and test something. Okay. We're going to make it. I'm going to tell you that we have changed temperature, tension in the membrane by pulling on the membrane, the ratio of the lipids in the sample, which could get us closer or farther from a critical point, the types of lipids in the sample to give lipids that have the dipole that was hypothesized, or to not have the dipole that is hypothesized. But unfortunately, very few of the theories actually make predictions in terms of these three knobs. Every single theory gives the same prediction for temperature, so it doesn't distinguish between any of the theories. So which ones are they? Two of these theories talk about tension. Same two theories talk about, would comment on the ratio of the lipids in the system, and one would talk about the types of lipids in the system. And I will cut to the chase, to the time, and tell you that I already said that number one is consistent with every theory. Number two is consistent with both of those theories that describe it. But number three is inconsistent with those same two theories that describe how it would change with the ratio of the lipids. Could it still be due to the repulsion due to the dipoles on the membrane? Well, the dipoles that are implicated in this system, when we remove those dipoles, we still see stripes. So, we don't have much time, so here we go. I already answered that question. How should you think about this system? There's no current testable theory that describes these specific stripes and dots. You could be like, oh, my life is terrible. We have very little theory to describe our data. Like, often we tell this story about how science works. Like, we have an idea and then we write a theory and then we test the theory, and I don't have a new theory to test. But the optimistic part is that, like, hey, students, this field is wide open. I mean, if you were like me and you saw that list of theories and you're like, oh, there's nothing more to do in this field, everybody's done this. Yeah, but it didn't explain the data. So go forth, write new theories, make new things that I can test, call me up and I'll test them. It'll be really wonderful to collaborate with you. And so that I'm gonna end. I'm gonna thank the stars, who are Chantal and Caitlin and Scott and Glennis and Alex. If any of you ever reviewed my grant proposal, I am so grateful. Thank you. I couldn't do it without the funding, and just fantastic collaborations. Candace and Ilya for collaborations with their group, and then for introducing me to their friends I can make more collaborations with. And you, thanks for the great questions. Thank you. Being aware that there may be people who need to go
make a
somewhere.
comment and then I need to go.
Okay.
There is a very different class of theory. You look up an old paper by Nigel Goldenfeld,
Okay.
which has to do with long range repulsion.
Okay.
And then you can map it onto the Rayleigh-Bénard convection, which has stripes.
Okay.
It's someone and Goldenfeld.
Yeah.
You can look it up. But
Okay.
it's basically if you have a non-local interaction, which can come from long range repulsion, then you can write down the usual phase separating equations. But you have to rewrite it because it's no longer like a bipolar.
So I am totally fine with that, but I want to know how I can turn it off. So I want to know.
Turn it off by changing, making the long range, somehow it's the range of interaction.
But yeah,
So if
but
there's
how
a
do I
repulsion-
test it? Like,
I don't know, that's why I was asking, can you change the width of the stripes by doing anything?
well, yes, we can change the width of the stripes by changing temperature, by changing tension in the membrane.
Yeah, so this theory was kind of made, it's basically maps onto the Rayleigh-Bénard or Swift-Horn, which are the lengths.
Okay. But if it only changes temperature, then it doesn't distinguish
Between
all the other theories.
all the other theories I don't care about.
Yeah,
I think this
okay.
is a very different class of theory.
Okay.
Because it's talking about, it's a whole, it's different. It's sorry, I have to run. But because what happens.
I know how to get in touch with you. It's okay.
What happens in these transitions is the critical point, and I think so one way of testing it, the critical point comes from fluctuations.
Uh-huh.
So it would have been a first order transition if there were no fluctuations. And if you put that into these field theories, then these kinds of stripes happen. They have to do with, it's weird, it has to do with the nature of fluctuations rather than things that we know how to control. So.
Okay.
Yeah, so that's it was a big focus in the mid-90s, I think, to figure out how phase separation happens in fluctuation-driven transitions.
Uh-huh. Yeah.
But then everyone's for that.
Sure. Okay. I'll see you later. So nice to think. Thank you for coming.
Take care, Sarah.